Hi team, welcome to EBH 1102, Lecture 3. This lecture we're going to look at the cell, organelles in the cell, and transport processes into and out of the cell. So just to give you a bit of background on the cell, it was first discovered in the late 1600s. And in 1832, German scientists insisted that all living things were composed of cells, and that cells arose from other cells. So that was a novel concept at the time, and which challenged the prior theory that organisms arose spontaneously from garbage or other non-living matter. Then the cell theory was developed, and cell theory states that the cell is the basic structural and functional unit of life, that the activity of an organism depends on the activity of its cells, that the biochemical activities of cells are dictated by the specific subcellular structures of a cell, and that continuity of life has a cellular basis. So in other words, all cells come from other cells. Just have a look at some cell size here. Here we are, here's our animal cell, that's us, sitting around 10 micrometers. Plant cell's a little bit larger at 100 micrometers. There's your bacterium down at 1 micrometer. A virus here sitting at around less than 100 nanometers. And molecules are sitting down at 1 nanometer. I'll look at the anatomy of a typical cell, generalized cell, and we're going to go through all these things. We'll look at the nucleus, the cytosol, or the soup of the cell, the plasma membrane, the part that surrounds the cell, the rough endoplasmic reticulum, which are sort of extensions of the nucleus, smooth endoplasmic reticulum, which is similar but doesn't undergo protein synthesis, the ribosomes, which sit on the endoplasmic reticulum and make protein synthesis, Golgi apparatus, or the warehouse of the cell, the mitochondria, or the powerhouse of the cell. There's the lysosome and the peroxisome. Those are the main things we'll look at here. We'll look at those in a little bit more detail right now. The microtubules as well we'll look at too. So we'll start with the nucleus, and that is the control center of the cell. Contains the DNA, which is responsible for providing the cell with its unique characteristics. There's a nucleus. Communicates with the surrounding cytosol via a number of nuclear pores, sitting right there. And the nucleolus produces ribosomes, which move out of the nucleus to positions of the rough endoplasmic reticulum, where they're critical in making protein. Next is the cytosol. It's the yellow part down here in the cell. That's the soup of the cell, they say. Just sort of the part where all other cell organelles reside and where most of the cellular metabolism occurs. The plasma membrane is the lipid membrane that surrounds the cell, controls entry of ions like sodium, potassium, and calcium. There's the endoplasmic reticulum, and that is a continuation of the nuclear membrane present throughout the cell. And under an electron microscope, some look smooth and some appear rough. The smooth endoplasmic reticulum is important in the synthesis of lipids and membrane proteins, while the rough endoplasmic reticulum, it appears rough under the electron microscope due to the presence of ribosomes on the membrane surface, which are responsible for protein synthesis. The Golgi apparatus, also called the warehouse of the cell, is a stack of membrane-bound vesicles that are important in packaging macromolecules for transport elsewhere in the cell. And the mitochondria is also known as the powerhouse of the cell and provides the energy a cell needs, or that ATP. Example of mitochondria sitting down there with the little crystals. The lysosomes contain hydrolytic enzymes necessary for the intracellular digestion, and they serve to kill and digest any bacteria that might get into a cell. Peroxysomes are responsible for protecting the cell from its own production of toxic hydrogen peroxide from lysosomes. And the microtubules are responsible for the movement of cell organelles, cell locomotion, as well, and muscle fiber contraction. So things like actin and myosin. There's the actin that's depicted there. Last section we'll look at in this lecture, just a short one. We'll look at the movement of substances through a plasma membrane. And these include diffusion, facilitated diffusion, osmosis, filtration, active transport, exocytosis, endocytosis, phagocytosis, bulk phase endocytosis, also known as penocytosis, as well as receptor-mediated endocytosis. We'll start with diffusion. Diffusion is defined as the tendency of molecules or ions to scatter evenly throughout the environment. It will always occur from areas of high to low concentration. Think, for example, a cut onion or an onion that's cut, and the sulfuric acid, which is the spray that's sort of released when the onion is cut. It diffuses through the air, and it doesn't take long for someone sitting far away in the room to notice that onion has been cut. And that is diffusion, moving from its high concentration near the onion scattering throughout the room. And I guess that's demonstrated in the little jar here, where the high concentration diffuses out into lower concentrations until it reaches some sort of equilibrium. Now, a molecule will actually diffuse through a cell membrane if it is lipid soluble, so if it's made of fat or soluble in fats, or fats are soluble in it. If it's small enough to pass through membrane channels, which might be actually in that plasma membrane, or if it's assisted by a carrier molecule. We'll look at simple diffusion first, and that's when something just actually goes directly through it, such as the double yellow dots right there. It's lipid soluble, so it's going to go straight through this lipid membrane right there and right into the cell. And things like oxygen, CO2, fat-soluble vitamins, and alcohol are examples. We also have facilitated diffusion, so some molecules, like glucose, for example, they're too large to pass through the lipid bilayer, such as this here, and they require an actual transport carrier, such as this here. It changes shape to allow the transport of that glucose into the cytoplasm. This is osmosis, and it is the diffusion of a solvent, usually water, through a membrane from areas of low concentration to high concentration. It's sort of the opposite of diffusion. So take, for example, here this example. We've got a high concentration here on the right side and a low one on this side. And because this membrane is permeable to these solutes, it goes straight through, and we see just an example here of diffusion. But in the second example, right here, we've got the same starting setup, but the membrane here is not permeable to these molecules on the right. As a result of that, the water actually changes. Water can go through that membrane until both sides reach their equilibrium, and the concentrations are similar. Osmosis. Filtration is the next one, and that's a process that forces a water and the solutes through a membrane or capillary wall via fluid or hydrostatic pressure. So just imagine like a coffee filter. You're pushing, the fluid has a hydrostatic pressure, filters out the coffee grounds, and the part that you want in the coffee comes through. Here's an example in the kidney. Here's the glomeralis, where the hydrostatic pressure is pushing through, and only the blood is retained, and the unwanted products are pushed through, such as the urine. So things in the urine like urea, creatinine, and uric acid, for example. Active transport's the next transport process, and I guess the remaining ones that we'll see here are all active transport processes. They're called active because the ATP is involved in this one. So it says here, similar to facilitated diffusion in that it requires carrier proteins, but it also requires ATP, as these solutes are going to move uphill against a concentration gradient. Typical examples, the sodium-potassium pump here, where you can see it actually pumps against its concentration gradient, pumping sodium in there with ATP. So we can actually see ATP is present as well. There's the ATP part. Exocytosis is also an active transport process, and you can see here it requires ATP. Here this is going to actually move substances from the cell interior into the extracellular space. So things like neurotransmitter release, like acetylcholine, will actually work that way. Endocytosis, sort of the opposite. It means provides a means for large particles or macromolecules to enter the cell. It's active transport, so it requires ATP. And there's three types we'll look at here in the next little bit. We'll look at phagocytosis, bulk phase endocytosis or penocytosis, as well as receptor-mediated endocytosis. Start with phagocytosis or cell eating. Here we have the cytoplasmic extensions called pseudopods. They'll actually flow around large particles, such as some white blood cells, for example. Well, actually, there's the pseudopod there wrapping around it. And here is an actual example. There's the bacteria here, this yellow part, and this is the white blood cell that actually is wrapping around that and ingesting that. So that's a phagocytosis in action. Bulk phase endocytosis, or also called penocytosis, could be termed cell drinking. And here's an actual another pseudopod actually occurs here. We have an infolding of that plasma membrane surrounding a droplet of fluid-containing molecules that extracellular fluid-containing molecules that the cell wants. So, for example, in intestinal cells that absorb nutrients. The last one here is the receptor-mediated endocytosis. This is the main mechanism for the uptake of most macromolecules. So in this situation, an external substance will bind to membrane receptors, and as a result, macromolecules are taken in. A typical example here is insulin, causing the GLUT4 receptor to translocate to the cell membrane, and therefore allowing the glucose to come in. So we can see here in the purple triangle, so there's the insulin, which hits the receptor, causes the infold there, and allows this glucose here, the green glucose dots, to be taken in this situation. And that's it for lecture 3, a very short one. But we'll continue on and expend a little bit more on lecture 4, which is quite a long one. There's some course compass homework here, chapter guide number 3, the cell, which should be completed to help you learn some of this material.